Professor Mayank Mehta, University of California, Los Angeles, USA. He will be speaking on the title Neurophysics of Space, Time, and Imagination. Professor Mayank Mehta is professor at University of California, Los Angeles. He is affiliated to the Department of Physics and Astronomy, Neurology, Neurobiology. He obtained his PhD from IISc Bangalore in theoretical physics, and subsequently he did his postdoctoral research in neurophysics in MIT, UC Irvine, and University of Arizona. His current research includes neurophysics of mind, memory, and information. One of the areas in which his team works to learn more is the method by which ensembles of neurons encode information about the physical world. Understanding how the brain perceives space-time and how these neural representations change as we learn is necessary for this. His group investigates how sleep affects learning using cutting-edge methods. He focuses on creating the next generation of artificial intelligence that is based on the human brain and non-invasive reversible ways to turn on and off specific brain regions. In addition, he studies how individual neurons respond to virtual reality and how information is processed by neurons as they sleep to generate long-lasting memories. His findings would not only offer a fundamental knowledge of the dynamics of neuronal ensembles, but they would also suggest brand new approaches to treating learning and memory problems. In order to comprehend how animals perceive space, he employs virtual reality. The theories put forth by Professor Mayank Mehta demonstrate how virtual reality might enhance healthy brain rhythms by changing neuronal dynamics, circuitry, and plasticity. His outstanding list of academic publications includes numerous publications in Nature and Science. Please welcome Professor Mayank Mehta. Thank you.
Thank you. So can everybody hear me well, I imagine? So let me get started. Thank you for inviting me here. Let me get started by introducing very briefly what is the question and why we should look at it. So let's get started. So in physics, we have the standard model right now of the universe. And these are all the particles that make up everything that we know. And recently, there was a lot of excitement because the mathematical formulation of how the universe works and the experiment for W boson changed. The difference was less than 0.1% between the current model, the standard model proposed by India, Pakistan-born Abdul Salam and Weinberg. That standard model has been working so well that when there was a 0.1% difference between the model prediction and data, it showed a huge sort of excitement. And that's the kind of work that I have been doing for many years. This is just one equation from my PhD thesis. So people say, well, the equations of the universe are very simple. That's not true. This is just one equation from my PhD thesis. And you can see this is not exactly simple. So the question is, can we come up with a mathematically precise description of the human mind, and how does it come about? And the standard answer that people give is that, oh, human mind is too complex. It has this amazing phenomena. We can never capture it exactly like the physics of the universe. The answer is no, universe is already complicated and people could do it. So what we need to do in order to understand the mysteries of the mind beyond simply meditation and worshipping some gurus is to actually think carefully about experiments and theory, mathematics, and then ask the right question. So I'm going to pose you the right question right now. All right, so let's get started. So we have these things that are going on. The world of standard model didn't come out of nowhere. It came from quantum physics and relativity, which came because of Newton's laws, which in turn came because of Kepler's work of Kepler's laws of two-thirds power law. What we need in neuroscience is something like that. And that came because there was somebody called Galileo who decided to go against the teachings of the religion, who decided to go against what everybody thought was obvious. You look at the sky, everything is going around the earth, so why even bother? He decided to do actually careful experiments. And what we need right now in neuroscience, or to understand the mysteries of the human mind, is to follow and approach something like this, where we don't simply look at our past and say we knew everything, there's nothing new to discover, or everything that we find is already known, but actually look at it carefully. And then be prepared that when we say this, we may meet the same fate as Galileo, who was killed because of his surprising discovery. So let's get started. So please stare at this picture and tell me when you see something. So just say, I see it when you see something. Can anybody see anything in this picture? Anybody see anything yet? Anybody? So this is an experiment that will show you that your mind can be changed by a simple image. So let me start telling you what is in this picture. So notice this is the outline of a Dalmatian dog. He has these spots on his body. This is his front left leg. This is his right leg. This is his face down there. This is his mouth. These are his ears. This is the one back leg. This is the second back leg. That's his tail. And that's a background. That's a tree here. There is a water port here. Everybody sees that? Many of you, as soon as I start telling you what to see, you will immediately start to see a Dalmatian dog. Even though they are just a bunch of black and white pattern, and just 30 seconds ago, most of you were not able to see anything in this picture at all. So the question is, what happened in your mind? What happened in your brain? So that while when you saw this picture, you were not seeing anything, you were seeing just something black and white and some dots, you are actually able to see some image. How does the brain create that? Clearly, it is not just what you saw, but you use some amount of imagination, partly aided by my suggestion. So, how did that happen? So, inside the brain, we have neurons. And these neurons, they create something called spikes. I'm trying to freeze one spike for you. So look at this. So this is actually a picture of an oscilloscope while somebody was activating one single neuron in the brain. And in human brain, there are 87 billion neurons. What is 87 billion? Number of people on this planet are roughly 10 billion. So the number of neurons in our brain are equivalent to about 9 planets size of Earth fully populated, which we don't have. Now, if you listen to just one person, if each neuron was a person, it creates a very complicated signal, and every now and then it creates this very large amplitude signal called a spike. Now, I have told you this spike is so loud in our brain that you can put a tiny little microphone of the right technology and you can actually hear it. On just an ordinary microphone and a speaker, you can hear it. As you see right now, this was discovered 100 years ago. Now, no matter how much you introspect about what your mind is doing, you will have never, even though I'm telling you this, you'll never hear spikes happening in your brain, even though it's a fact that all neurons actually generated spikes like this, and they sound pretty much like that. So I'll play that once again. So the magic is right there. How is it that every element in the brain creates such strange language called spikes? That's magic number one. Magic number two is, how do so many neurons create this magic so that we, the possessor of these things, can actually not see these spikes? We can't see this. We can't see the spikes. We can't hear it unless somebody else shows that that's the spikes from somebody else's brain, not our own brain. We ourselves cannot perceive what is going on in our brain. In fact, there is a lot of emphasis on understanding what is consciousness. The real fact is that 99.99999% of what's going on in our brain, we are actually unconscious of. The real problem is unconsciousness. How come we cannot feel the most basic thing that an ordinary amplifier can hear, we ourselves cannot hear? Instead of that, we hear the world and we see the world as if it is real. So how does that work? So that's the question. How does the brain create intelligence? This is the human body, and the brain is just that size inside. And still, people say, well, the reason human brain is able to do these things is because it has 100 billion neurons, whereas a computer doesn't have that. Well, that's not true. People have recently made computers with 100 billion elements, and it doesn't have any ability to create intelligence like human. So number of elements in a computer, number of transistors, which also make zeros and ones, they are already reached that level and they can't do that. People think maybe the reason human brain is special is because it is very, very fast. Because these spikes that you heard, they are only 1 millisecond wide. 1 millisecond is how much? That's 1,000th of a second. Isn't that amazing? Well, that's not true. The computer clock runs at 10 raised to 9 hertz. That is 1,000,000 times faster. That ordinary laptop that you have right now, which many of you have, is running 1,000,000 times faster than the human brain. And so is this computer with 100 billion neurons and run 1,000,000 times faster. Still, is not able to do what human brain does, which is to see a bunch of dots and say, here is a dog in it. But there is something interesting that our brain does. Our brain consumes 20 watts of power. Now, what is 20 watts? Our brain consumes 1/5 of all the energy consumed by the rest of the body. The brain is pretty small. It's just about that much. About 3% of the body's weight is the brain. And even though the brain is 3% of the body's weight, it consumes 1/5 of all the energy. Instead of 3%, it consumes 20 to 25% of all the energy. And even though it consumes so much energy, it's only 20 watts, less than an ordinary LED bulb. Whereas this computer uses 20 kilowatts. So it's, again, 1,000 times more energy. So there is something special about human brain that can be understood from principles of physics. Because in physics, we know what takes energy, what is energy, is it conserved, what forms of different structure energy can take. So we want to understand how come brain takes so little energy, even though it is so much slower, and at the end, it is able to do something that some computers, which are way better than human brain, cannot do that. So there is going to be something about the energy consumption. In addition, what I will show you is that our brain is actually not only very slow, running only at 1 kilohertz, it's also very sloppy. When you show the same stimulus 2 times, the brain's responses are very different. Not like a computer, when you say 2 plus 2, it always says 4. When you show a picture, it creates the same response. Our brain creates different responses. And our brain, unlike computer, feels very sleepy. Computers don't need to sleep. We need to sleep. Why do we need to sleep? People may think when we sleep, we are asleep to save energy, but that's true for rest of the body, but the brain continues to burn as much energy during sleep as when you're wide awake. So on one hand, the brain has been developed to save energy so much heavily. On the other hand, brain keeps using energy for 1/3 of our life when we are sleeping 8 hours a night. That's a third of your life. Why does it use so much energy during sleep? So let's get started with it. So how do we ask these questions about what the brain is doing? We can't listen to individual neurons in human brain because, well, in order to do that, we have to drill a hole through the skull, without which you cannot measure what a neuron is doing. The sounds that I showed you were obtained by drilling a small skull, even though under anesthesia. So in order to do that, we have to use small animals like flies or mice, where we can put some electrode and measure what's going on inside the brain. But now comes the next problem. A mouse or a fly is not a human being. Then the question becomes, does a mouse or a fly or a bird have intelligence? Or is intelligence a unique ability of human beings? Clearly, that's not the case, that animals are not intelligent. If there is a fly in the room that's bothering you, try to catch it. And with your 100 billion neurons, you'll have a really hard time catching the fly with your bare hands. It's going to escape. So clearly, you can't match the fly's ability, let alone you're not able to fly even as the fly can. So what's special, and how can we ask what is intelligence and what is imagination? How can we listen to single neurons? So what I'm going to tell you is, after my research, what I have learned is that there is one, or rather two, concepts that are universal, that all the animals in the world, despite the different brain and different evolution and different behavior and different species and different genomes, all the creatures on the planet agree on two things always. Those two things are space and time. Now, what do I mean by space and time? Everybody has to agree. This is a bird that flies from North Pole to South Pole every year without any GPS, without any complicated device, on its wing, doesn't get lost, lands on the same place. In addition to being able to do that, which we of course cannot do, we can't fly, we can't navigate so much distance. But in addition to that, on the way, as it flies from North to South Pole, it gets hungry and it dives and picks up a little bit of fish that is swimming near the surface of water, right there. Now, that is where magic happens. Why is that? Because while we think that space and time are real, our brain is completely imagining what they are. I can ask you right now to lift your hand and touch a point in front of you 20 centimeters away, and then I can ask you what is in that point right there. Can you smell that point right here? You cannot smell it. Can you see it? No, you can't see that point either. You can see the walls all around you or people around you, but you can't see the empty space between you because it is empty. Then I can ask you the question, what did you mean by you wait 20 seconds? How did your brain know what is 20 seconds or 5 seconds? These concepts of space and time are independent of human language. If the bird's ability to know exactly where the fish is and to catch the fish exactly at the right time that it came near the surface, and the fish's ability to say the bird is coming, let me hide it, that precise time to say bird is going to land here and let me run away, if those two things did not match 100%, and when I mean 100%, I mean 100%, either the birds will eat all the fish in the oceans because the fish are not very, very smart about figuring out how quickly the bird is going to come and where it's going to land and at what time. birds will eat all the fish and life will come to an end because then birds can't eat anything. Or the birds can't eat any fish. The fish are just a little too smart for birds. Birds can't find any fish. All the birds are dead. Once again, life comes to an end. So we, all the creatures on the planet, at all moments when we are awake, we are constantly using the idea of what is around us, how far is it, how long will it take me to grab it. These are all made up quantities, and even though they are made up quantities called space and time, because made up meaning we can't feel the space and time, we feel it is real. We have a strong feeling that there is space between us and the next person, but there is empty space. You can't see or feel it. So how does the brain do that? So that's what we do. We try to understand deep questions about how do we perceive abstract ideas such as space and time. We look at what goes on inside networks and we figure out how individual neurons make that happen. So in the brain, as you know, there are 100 billion neurons. These neurons are connected by small little synapses, and there are 10 to 20,000 synapses per neuron. So that's another magic. Ordinary cell in your body, such as on your fingers, on your muscle, every cell touches on an average about 10 other cells. Inside the brain, it has come up with a unique machinery not found anywhere else in the universe, let alone in the body, where each neuron listens to 40,000 other neurons through these synapses. Imagine listening to 40,000 people speak at the same time. You can't understand what two people are saying if they both start speaking at the same time. Each neuron is listening to 40,000 other neurons all at the same time. So there is some magic going on there. So the brain is not only very interesting in this mathematical sense, but its circuit is very, very complex. And at the bottom of the brain is the eye. Then there are many brain regions. And if you want to go to a brain region that's farthest removed from the periphery, you reach this brain region called HC or hippocampus. That's in one of the deepest regions of the brain. It's also one of the oldest regions of the brain called hippocampus. That's the part of the brain which is unfortunately sensitive to many diseases, such as Alzheimer's, autism, epilepsy, PTSD, depression, schizophrenia, and so on. And just the cost of treating Alzheimer's is $5 trillion, astronomical. Lots of people have tried to cure these diseases. We have been able to cure a fever or a stomachache or all kinds of cancer. COVID, even we human beings were able to cure, but people have still not been able to cure Alzheimer's, which is one of the oldest diseases, or dementia people can't cure. And the way that happens is through understanding how this part of the brain creates these abstract ideas. So I'm going to play a little bit of a video clip of how that part of the brain
which I'll say
operates.
very little about because I'll leave it to the Moses to describe
So
that.
here is the Nobel lecture by this person named John O'Keefe, who discovered what these individual neurons in hippocampus do when the animal is running around. And for the discovery in 1971, he got the Nobel Prize in 2014. So let's listen to him describe what those neurons are doing and how they are creating space-time perception.
In much greater detail. So let's go through and look at these cells in a bit more detail. So first there are the place cells.
So he's going to place a rat. Imagine he's going to create an animation of a rat that will be placed on this big
If we
disc.
place an animal, this is a cartoon made by my colleague Julia Krupich. If we place an animal into a circular environment, we watch where it is using an overhead camera, and we also register where it is when one of these place cells fires. What we see is, by and large, the cell is silent over most of the environment, and it's only when the animal goes into the part of the environment which is preferred by that cell, its place field, that the cell fires. And so you can see all of these red dots are adding up, and what we typically use is a heat map to represent the firing rate of the cells. So the heat map shows that this cell fires with a maximal firing rate of 7 hertz here, and successively lower rates as you move away from the center. And this is the typical type of way in which we represent these cells. If we look And one thing that I should point out is that if an animal runs around in an environment like this, in an open field, it turns out that it doesn't make any difference which direction the animal runs through the place field. If it runs north, the cell is happy to fire. If it runs south, it's equally happy to fire. It looks as though the cell is representing the location and not something about the sensory impressions that the animal is getting in that location.
So this is a major discovery. This part of the brain, when people put tiny electrodes and listen to individual neurons, which is quite a difficult challenge, which these people overcame in 1971, they found that individual neurons seem to pick up an arbitrary location in space and they can make a map. And we can do that too in our lab since that discovery. So here's the data recorded in our laboratory. And a mouse was simply running around in the maze, picking up little bits of food. So this is data from our lab now. And those crackle is the individual spikes that you heard. And this trajectory I'm showing you just for the sake of your visualization. There is no trajectory. This just shows you the mouse's path. And what you'll see, now we speed up the simulation. And what you see is that even though the mouse is going all over, and this simulation we are running much faster than actual data. This is a whole half an hour long experiment in which the mouse does all sorts of things. It sits down somewhere, eats some food, takes a nap, scratches himself, does all sorts of things. But the neuron generates spikes only here. And as I can see, the aspect ratio is messed up. So this doesn't look square to you. But trust me, the screen is a square screen. There's just some audio-visual glitch that's causing this to look very distorted. And when the mouse is running around, it has all these visual cues on the wall. There is a spiral somewhere. There's a big sunflower on another wall. There is some other target on another wall, another mathematical object we created by hand. These are all mathematically created objects in the lab. These objects never existed in the universe until we made it. So the mouse is definitely seeing them for the first time. And even though it's seeing them for the first time, it is able to make a map of that space to say this neuron fires here, a different neuron fires here, and so on. And this classic idea is that these neurons do not care for what the mouse is looking at. These are abstract representation. The neuron's activity is independent of what the mouse is looking at. The question is how? How do we know that that's what these neurons are doing? How do we know that that's what they're not doing and not that the mouse is smelling something which is on the ground, which we human beings can't smell because we are 1 meter away from our ground. Our nose is 1 meter from the ground and our nose is not that sensitive. What if mouse was smelling something? Or even worse, what if mouse, which has these whiskers, which are extremely sensitive, we don't have such whiskers, we have mustache and beard, but they don't have sensitivity. We can't move our beard and mustache like a mouse does. What if the mouse was sensing tiny little bits on the ground? In fact, many people have said that mouse vision, because they are nocturnal creatures, its vision is not very good, which is true. It's not like human beings, which rely on vision. So maybe the reason the neuron fired here was not because it was a sensory, it was some abstract space as we imagined it is, right now as I give you the example, but because the mouse was just smelling something. How do we know that's the case or not? So this is a fundamental question. In some sense, the equivalent to say we are seeing the stars move around and planets move around the Earth before Galileo, and we thought, yes, everything goes around Earth. That sounds right. Is that really so? So we want to start doing experiment like Galileo and ask the question, this neuron's response does seem like it is just a place that is abstract, but is it abstract? And then we ask the question, like Kepler and Newton, as to if the response is abstract or not, how did it come about? What is the mechanism by which it came about? So let's get started. So how do we do that? Well, we thought one way to make sure that the mouse behaves like human being is to make virtual reality. So just like Galileo made sophisticated telescopes with which the Galileo is able to look at moons of Jupiter and figure out things are going around other objects, not just Earth. We wanted to make an object where all these other stimuli are removed. So there's a big styrofoam ball on which the mouse, or the rat in this case, runs. The movement of the styrofoam ball is picked up by this microcontroller. That microcontroller sends the image to a virtual reality engine. All this was built by these brilliant set of people in the lab. And then that image goes to a pico projector that is hidden above the rat's head. That image then goes to this polished mirror that astronomy friends provide us. And then that image goes all around the rat so that he's immersed in virtual reality. So here's a picture of the rat wearing a little harness, standing in virtual reality, looking at all the world, and he can see his own shadow. This is the ball on which he's standing. This is a little platform around him. So he doesn't feel nervous. We want him to be not stressed out. We want him to be happy. And this little harness, like a tuxedo, he can slip out whenever he wants. So, and here we can show the little image of how that looks when some experimenter moves the ball by hand. And you can see, even though the camera is outside where everything is distorted, you see a compelling sense of something is going on all around. Everything looks undistorted from the point of view of the rat at the center of this world, right there. With a webcam outside, things look distorted. So now in this virtual world, the rat cannot hear, he can smell things, but what he smells on the ground is constant because he's always at the same place. What he touches on the ground is also constant. It's always the same spot. Only thing that changes are the visual scene. And the question is, can he figure out where to go in this visual world and create a concept of space? Now this virtual space is 100% something that no animal had experienced before, let alone rats. Let's see if the rat can perceive the visually created space as real. So things will look distorted and dull because rat vision is unlike human being. They like very dull light. Bright light is too much for them because they are nocturnal. So the scene will look a little dull, but let's watch what the rat does when he has to run around in virtual reality. So that's something now one can have a human being wear virtual reality goggles, maybe in a clinic or maybe elsewhere, and the rat runs here. So let's see what the rat does in virtual reality to do a little task. So he has to run and go underneath this imaginary chandelier to get a reward. And when he goes there, he gets a little bit of sugared water from the bottom. So now while he's drinking sugared water, the chandelier goes somewhere else. Watch him. He looks up and down as if it's real and checks underneath it to see if it got reward. Now, the next trial is even more difficult. The chandelier has moved behind him. He has to look for the chandelier. He sees the floor of the virtual reality, which has a different texture than the table on which he is, and he goes under the chandelier once again. Now I'm going to show you one more trial in which he did this. So there is one more trial where he does this. So we keep going forward. Again, it's a very hard thing. He has to look for it. And he finds the chandelier, he sees the floor, even though he can't feel the floor, and turns around and goes there. So he can do many, many trials in this task, which is a perfectly normal task to say, go underneath some tree and find a fruit, or go underneath the chandelier and find some juice. All creatures do that all the time. He's making up this space using only vision. He's not going anywhere. He's in the same place. So it's a unique experiment where you are going somewhere, but physically you are in the same place. And yet it's not like watching television where you're sitting somewhere and an image is going. It's not that. Here you're going somewhere because of the movement that you made. You walk forward, you walk backward, you walk left, you walk right. And that's what caused the movement. And that's why it created a sense of the world going around you and you are in it. It's a made-up world, completely fictitious world, made only of light. Now we can ask the question, how does the brain create a representation of space using only vision? And is it an abstract representation, Because, or is it that the smells and the textures were playing a role, which of course for human beings don't play a role. So to control for this experiment in science, we want to do control experiments. We ran the rat in RW. RW stands for the real world, which had the same stimuli as the virtual world, same psychedelic stimuli that I showed you in the very first slide where the rat was running. And then we made him run in the virtual reality with the same colored walls with spirals and target and flowers and so on. And now when he runs around in the real world, you can see many of these ellipses are different neurons. Each ellipse, each dot is a spike. The crackle that you heard is the sound of individual spike. And you can see on this electrode, which is a fancy electrode developed by many scientists, including John O'Keefe, those crackles, this electrode can listen to about 10 different neurons at the same time, which are all active at different places in the real world. So it shows that everything is normal in the real world with this electrode. When we look at the same set of neurons with the same electrode in the same rat, with the same visual cues, what you see is that many of these neurons simply shut down. This neuron is simply shut down. That neuron right there is shut down. This one has kind of appeared, but it's a different neuron. This big ellipse of a neuron shut down. That big ellipse here shut down. So what you see here is something remarkable. And we did this experiment across many rats, that 60% of the neurons in this part of the brain are shut down in virtual reality. Now, many of you have felt many times that, your brain is too active, your mind is too active, and you want to do meditation or something else to calm down your mind. You want to focus on a candle or something else. You don't really know if your mind is getting shut down or not. There is no drug right now available to any human being that you can take a pill and shut down the brain. Some drugs are available, such as GABAergic drugs, but if you shut down your brain more than 2% using a drug, you will die. Because when you take a drug that shuts down the brain, it also shuts down the part of the brain that controls your heart. And if your heart starts to slow down, there is not enough oxygen, you die. Same thing with your lung and so on. So drugs cannot do this, whereas this fancy new technology, which we never imagined will do this, is able to shut down the neurons in hippocampus by 60%, just like that, which no amount of meditation, no amount of drugs can actually do. Now the question is this good or is this bad? Turns out this is a good thing for patients which suffer from epilepsy or early stages of dementia or early stages of Alzheimer's. When patients have epilepsy or early stages of Alzheimer's, the neurons in that part of the brain become too active. When they become too active, they consume too much energy. The energy is not available. So those neurons start to die. And then people's memories suffer. So if we can simply shut down the activity of the neurons, this part of the brain sucks up less energy. Potentially, we have slowed down or even stopped this part of the brain from getting long-term damage. Is this true? We are going to find out. We have filed for a patent. We want to do this, but none of this we could have imagined before we started to do this. So now let's go to the bigger question. What do the neurons do when the rat's actually running around, the 40% that are actually active? So now the rat's going to run in the same virtual reality, same walls, but it's virtual. The neuron is active, and you see this burst of activity. Now it's a different bird in a different place. So what you can see here is that the neuron is active everywhere. There is no place at which the neuron is active. But if you paid slightly close attention, so first of all, before I tell you the next surprise, is that even though if I were to ask you, tell me where you are in this room where you are sitting, you will say I'm 4 meters from the door to my right and 2 meters from the screen in front of me. And with those two coordinates, I can uniquely localize you. And that's how people thought that the brain localizes itself. But in this virtual world, where mouse was localizing itself only through vision, we know 100% because the world only contains vision. In that world, the mouse neurons were not able to localize himself at all. They formed a spaghetti. They were firing randomly. But so that starts to challenge the Nobel Prize winning discovery that neurons create a map of space by triangulation with respect to landmarks. This shows the mouse is actually behaving by triangulating with respect to landmarks using vision, but the neurons have no map. But if you look closely, and I'm now going to play this again for you because it's such an important thing, whenever this neuron is active, it has a certain specific rhythm. And the rhythm is fascinating, and the same rhythm keeps repeating many times. The rhythm has a motif that's about 2 seconds long, and within that is an interesting periodic structure. If you're musically minded or you know tabla a little bit well, you'll see the rhythm is about 8 times a second, which a tabla player can easily play. So let's listen to that rhythm, which is 2 second long motifs. Oops, let's go here. Yeah. So take a listen. There's a 2 second long motif. Sounds exactly the same with some variation. We're going to speed it up. So notice whenever it was active, it is the same rhythm. In reality, what this neuron is doing is actually representing time. That's what it's doing. So the brain actually activity in virtual reality resembles like what you see in Alzheimer's disease. This is the activity pattern of a neuron in the real world that got the Nobel Prize. This neuron is active in only this place. All the other area where the mouse was walking around, the neuron was not active. So it's not as if the mouse brain was damaged in our case. It's not as if our electrodes in the wrong place. Everything was working fine in the real world. Once you put the mouse in virtual reality, 60% of the neurons shut down and the neural activity pattern started to look very abnormal. Instead, the neurons started, instead of encoding space, the neurons are encoding these brief segments of time. You can see these segments right here. There's one segment there, there's another segment, third, fourth, fifth, sixth, and so on. So that starts to go off. And in fact, several people recognize our work. In an editorial in Cell, it says that our work challenges the decades-old understanding of how brain creates abstract idea, and our work will lead to the next Nobel Prize sometime soon. So this has been recognized by many people, and one can now use this technology to do a lot more. So the question is, and I will end after this, so question is, what is the brain doing in virtual reality? Because mouse clearly is able to walk around. So now the mouse is again going to run in virtual reality, but watch what happens. So he's still running in virtual reality, but something funny happens. So that's one motif, like you heard before, in the real or virtual world. That's the next motif right here. Also, like before, seemingly random, is a third motif. Notice what we did. Instead of the mouse walking around randomly in this world, he was walking always in a systematic fashion. When he was here, he always went forward. When he was here, he didn't go in this direction, he didn't go in that direction, he didn't go in that direction. Whenever he was here, he always went left. When he was here, he always went northeast. And when he was here, he always went south. And because his behavior was systematic, the brain was able to pick up this statistical regularity in the world and say, in this case, I know I have to only count distance. It doesn't matter that I'm walking on an equilateral triangle. I'm just going to keep track of distance, regardless of where I am. So now this starts to reveal what this part of the brain is doing. If you are recording the activity in visual cortex, it will of course look very different, because here you are walking toward this weird sign, here you are walking towards the target, and here you are walking towards the flower. So the visual part of the brain will have completely different responses based on in which direction you're walking. But in the deeper part of the brain, hippocampus, neurons have abstracted out the relevant information and said, I don't care whether it's a triangle or a spiral or a target. I just care for how much distance, and then I get the reward. That's all that matters, is to get the drop of sugared water, and that part of the brain figured out an intelligent operation to say, even though the vision looks very different when I go this way, the scene looks very different than when I go this way or that way, the neuron was able to abstract that out and create from this changing vision an invariant concept. So that's what it's able to do. So I end on this interesting note that we started to understand what the neurons are doing. Since 1971, the idea was that neurons create a concept of abstract space or a physical space, like a Newtonian space, which is a location of oneself with respect to the world. In using vision, we found that actually, if you look more closely, these neurons are counting distance if the paths are systematic. And if the paths are random, the heart of everything that is being made is actually, neurons are actually keeping track of time. And this 2-second period of time, in one case, is being morphed into creating a concept of distance and direction. And in another case, it's being morphed to say, this is the place at which I have a little bit of smell or a texture. So same internal trick that the brain is doing is being put together to create these widely different ideas. And there is a lot more which we have done after this, but I will just stop here. There's recognition from peers. This paper, when it appeared, people wrote editorial in Cell saying this study has uncovered a new level of complexity in the firing patterns of neurons in the hippocampus that ultimately will give us a deeper understanding of its function. There may be another Nobel Prize up the road for whoever makes this discovery. So all of us in India, since we are very deeply interested in the brain, we have a challenge to figure this thing out. And now, before I end my story, I want to use that experiment that I did on you once again. So tell me, what do you see? So for the last 45 minutes, you were not thinking about this dog at all. You were thinking about the complicated information that I was giving you. And still, now your mind sees this immediately based on one experience. So once again, that goes on to show that the same thing is going on also in the rat brain and hippocampus. And there are other stories that we have about where that happens. And all that stuff happens inside the dendrites, which is a long story, which we can listen. And maybe I will end on you allowing you to basically listen to a part of the brain which is nobody else has listened to before, which are called dendrites, where the memories are stored. So that's how that part of the brain sounds. So notice the sound is very different than what you heard before. That sound is very deep, unlike the high frequency sound. And these are the spikes generated in the part of the brain that people thought is the dark matter of the brain. It doesn't generate any spikes, which is 99% of the brain. So there is still more exciting stuff coming along, but I will stop here and take any questions that you may have. So let me ask the second question first, then I go to the first question. Notice I did not say that this is time perception in rat. I only stated this single neuron, one neuron out of 80 billion, or in case of a rat brain, 1 billion. 1 neuron out of a billion was keeping track of roughly 2 seconds. I have no idea what the rat's mind is doing. When Aristotle talked about time, that was about the mind. Mind is the result of complicated pattern of 87 billion neurons. So to put it in perspective, what I didn't tell you is that while one neuron is keeping track of this, like an hour hand or a minute hand, which is 10 seconds, and seconds hand, which is 10 times a second, one neuron is keeping track of time at the level of 2 seconds. Another neuron right next to it keeps track of half a second. Third neuron, 4 seconds. Fifth neuron, 10 seconds. So different neurons are simultaneously keeping track of different time. Each neuron has its own distinct motif. It's like an ensemble of 80,000 or in case of a mouse brain, an ensemble of roughly 1 billion musicians, each musician playing its own motif, which is different than other musician. So this is a whole different thing than time perception. This is something beyond my mind can comprehend, how to think about it. So that's the first part. The answer to your second question, what is so-called present, how thick is the present, actually is different for different neurons, very different. Now to the first part of the question, what is the molecular basis for this rhythm and so on? So we have been doing a lot of work on that. Requires not just biology, but solid amount of math. And the math gets very complicated even to understand just one simple neuron getting input from many neurons. And we recently made progress in simplifying this time creating circuit. What we discovered is that when you anesthetize an animal, now the animal is definitely unconscious, but it's alive. In that case, all the neurons synchronize their rhythm. All the neurons have exactly the same rhythm. And that rhythm is very simple. 2 seconds on, 2 seconds off. 2 seconds on, 2 seconds off. So the first challenge is, how does the brain create this rhythm, at least under anesthesia? So anesthesia in my mind is like the inclined plane or a pendulum, which is frictionless. Everything is simple. Once you become awake, it starts to do very complex things. So first, good news, even in an anesthetized animal, which is unconscious, the neurons generate the same roughly 2 second long rhythm, and all the neurons synchronize. So this, we thought, should be possible to mathematically solve, because we know what's going on to the neurons. We have the equations. We discovered this phenomenon in 2004. This is my most cited paper. And then it has now taken us 20 years, and now we have come up with the most simplified circuit. And that circuit says, I just need one excitatory neuron, one inhibitory neuron. There are only 2 conductances. And now in black, you have the experimental data, and in green, you have the theory. And you can see the experiment and theory will match extremely well. So it has taken us a long time, not just us, a lot of people are working on this, to come up with a simplified description of how you generate this 2 second off, 2 second off, but there is a lot of variability you can see. It's not always exactly 1 second here. Sometimes it's long, sometimes it's very short. So this theory can now do that. Now, the good part about the theory is that once we can match the theory to experiment, like we do in physics, where the theory is not just qualitative theory, the theory matches experimental data 100%. In fact, it gives you a short shift as well. When the theory and experiment match, then you can take the theory to the place where you cannot take the human brain directly because the animal may die, including waking up the brain. And that's when we see amazing new phenomena, which will appear in print soon enough. This paper is on bioRxiv. Krishna Chaudhary is a brilliant student. He developed this simplified mathematical theory. Before that, the theory was developed by another bright student, Maryam Gorbani, who is from Iran. She developed this simplified theory, which gives you very regular oscillation, 2 second on, 2 second off. Now, Miriam and Krishna had developed a theory of how the oscillations or the rhythm becomes more variable. And then we are going towards the next thing, which is quantization. So it starts to get more interesting once you start to combine different brain regions together. This circuit starts to generate very fascinating set of phenomena, including chaos and even quantization of time. So this is a long journey, but this is on the left-hand side is experiment. Right-hand side is theory. They look very similar, apart from color, which we have intentionally used different color. And notice this quantization going on. That's in the experimental data we could observe. And we find the same thing here. So it's a long story. I didn't tell you what happens with those oscillations during sleep. But what we find is that when those variable oscillations are happening during sleep, the brain is actually reorganizing memories to actually remove irrelevant memories. And that's another long story. But let me stop there and answer any other questions. Yeah. So again, these are new mysteries which people didn't know because the theory is just becoming available. But here is what happens during sleep. A little bit. Again, this is still too early. We are in the days of Galileo, and the question that we are able to ask are like, what is a black hole? In mind, that's a weird thing. We can't ask that yet scientifically. But here is something that work of a lot of people has shown. So it turns out that when we sleep, individual neurons are oscillating like this in their activity. They become active for 2 seconds, silent for 2 seconds, active, silent. They behave like a two-stage system, or as we call in physics, like a spin. Now, more interestingly, as the neurons do this 2 second on-off for a long time, there are other cells in the brain besides neurons. These are called glial cells. Those glial cells start to shrink. So here's the neuron, which has this tree-like structure, and glial cell, which looks like a fist. The glial cells shrink, and as a result of it, there is now a gap between the neuron and glial cells. And now the neuron starts to dump out all the metabolite. Just like when we eat food and oxygen, we produce waste. Neurons, when they consume all this energy, they produce a lot of waste product. These are large molecules. Those molecules have to be dumped out of neuron. But brain is tightly packed. There is no room for dumping out those molecules. So what happens is that in that case, the glial cells shrink and the neurons dump out those metabolites, and those metabolites that go through our spinal cord, and then we release it the next morning. So when we don't sleep well, that process gets messed up. And in that process, the metabolites are, of course, taking care of making the memories, removing the memories. That gets disturbed. And that's how memory doesn't get integrated. That's a short version of what happens. There is all sorts of other things happen, such as brain does time travel during sleep, and it runs through things very quickly and so on. But that's another story. First of all, I don't believe in magic. I don't believe in some complicated magical powers or something. The fact is, we don't know. We don't know how is it that the birds can go from North Pole to South Pole without GPS, without getting lost. How do so many billions of birds do all this stuff every year, which we human beings can't do? And the bird brain is only, 1 gram. So how does it do that? There are people who have ideas about how that happens. For example, birds can sense temperature on the ground when they are high up based on whether there is a drought somewhere or not. The nature of air currents will be different. You can see in the morning when vultures and kites are going in circles, they're using the air current because the air on the ground gets hot. The air above is cooler. The hot air from the ground starts to rise, and the vulture wants to minimize energy. So it rides this rising current of air to go up in the air. So the birds can perceive where is the hot air coming up. So it can tell from long distance that this area has drought and there is no hot air going up. Whereas some area, there is water. If there is water, the hot air is not going to rise as much because water heats much more slowly. Why? Because water has lower specific heat than ground. Well-known thing, that's what creates a breeze near the ocean in two different directions. So they could be sensing that. And without going anywhere, they can sense that there is this water coming up, the air currents, and they can go there. So that could be, but again, somebody has to look at it carefully from a scientific point of view rather than imagining that there is some mysterious ability birds have. There is no mystery, just pure science. We have to look at it carefully. All right. If there are no further questions, then thank you all very much.
Sir, thank you very much. Yes. I would like to extend our gratitude for you for joining us today and sharing your vast research, knowledge, and wisdom through the marvelous presentation that you gave. Thank you once again, sir.
Thank you. Thank you, everyone. Enjoy.